The Indian River Lagoon is one of the most biodiverse estuaries in North America. This biodiversity translates to incredible chemical diversity, which is what I'm interested in studying. More specifically, I research the specialized metabolites or marine natural products produced in these organisms, which can be used in drug discovery. The goal of my dissertation research is to identify these marine natural products which reduce levels of a protein called survivin in cancer cells, and therefore represent potential anti-cancer drug leads. Survivin is a protein that is upregulated in cancer cells and is correlated with poor patient prognosis. The Wright and Guzman labs here at Harbor Branch developed an experiment using the imaging instrument pictured in Figure C to measure survivin levels in cancer cells in response to treatment with marine natural products. You can see here the survivin protein is labeled in red and high levels are expressed in normal cancer cells in Figure D. After treatment with an active compound in Figure E, survivin levels are reduced, which is what we want to see. From this assay, an extract from a tunicate found in the IRL, Eutostoma olivaceum, pictured in Figure B, was identified to be a potent survivin inhibitor. I purified this compound from a complex mix of chemicals found in the E. olivaceum extract and determined the structure of this pure active compound. The next step was to identify how this pure compound was acting in cancer cells to reduce survivin levels. If we consider the central dogma of biology, it states that DNA makes RNA and RNA makes protein. So, if we are seeing a decreased level of survivin protein, we may classify that mechanism into two broad categories. Option 1, the survivin simply isn't being made, meaning DNA isn't transcribed to RNA, and we can measure that with an experiment called RT-qPCR. Or option 2, the survivin protein is being made, but it's being targeted for rapid degradation, resulting similarly in lower survivin levels. The results of my RT-qPCR experiments in the bar graph show that my E. olivaceum compound, labeled 41102, did not reduce transcription of survivin mRNA, so we can conclude its mechanism is not option 1. But when I look at the degradation rates of survivin protein to the right, there is a statistically significant difference of about 50% at the 12-hour time point for treated cells compared to control. This suggests that my pure compound from E. olivaceum is acting downstream to target survivin protein for increased degradation. There is still a lot of work to be done to determine exactly how this compound acts in cancer cells, but I believe this data is a step in the right direction. Thank you.